woodlands for wildlife. For 40 years, our organization has been dedicated to empowering Vermont landowners and others with the knowledge, tools, and community support needed to manage their woodlands sustainably and enhance wildlife habitat. We do this through our hands-on trainings, educational workshops like this one, and our peer-to-peer outreach. First, we want to go over a couple housekeeping items. We ask you please have yourself muted to minimize all background noise. You're welcome to have your cameras on or off throughout the presentation. And if you have any questions, please type them in the chat and our presenters will hopefully be able to get to them at the end as time allows. And of course, at Coverts, we're all about connection here. So if you want to start, you can say hi in the chat and type out where you're from. And without further ado, we are having here today with us the Vermont Forest Parks and Rec Forest Health Specialist, Savannah Ferreira, and Restoration Ecologist and Educator, Jess Rubin, and they're going to share with us today about the wonderful world of fungi in Vermont and all about what's going on there. If you guys want to take it away.
Thanks. So, yeah, hi, I'm Savannah,
Hi, I'm Jess.
and we're two members of the Fungal Scientific Advisory Group, and we're here to talk about the fungi of Vermont. So first, starting off, what are fungi? Fungi are not plants, like a lot of people assume. They're actually more closely related to animals. Although they do have a cell wall like plants, their cell walls contain chitin. They're absorptive heterotrophs, so they externally digest their food. They usually secrete enzymes that break down food in their environment, and then they absorb those back into their structures, as opposed to plants that make their own food, and then animals which internally digest their food. Many reproduce using spores and spore-bearing structures compared to seeds or zygotes, and many are made up of hyphae. Fungi can live freely in soil and water, and then also form a spectrum of relationships with other organisms. So why should we really care about fungi? Well, the short answer is that they're a keystone kingdom in our environment, and they provide a suite of services, which include ecosystem services, as well as symbiotic relationships. Ecosystem services are usually the direct and indirect benefits, typically in the context of human benefits, and they're generally divided into these four categories. So we have supporting, which is food and shelter for other organisms, helping maintain biodiversity. We have provisioning, so providing a service like food and medicine. We have cultural services, which is increasing recreation and tourism, enhancing aesthetic appreciation, spirituality, and again, medicine. And then lastly, regulating. So this is soil stability, it's carbon sequestration, drought resilience, and just overall ecosystem stability. These are just a few examples. There's really far too many to actually put on this screen. But wherever fungi are present, well, fungi are present wherever organic matter is present. So they're really ingrained in all of these services. Additionally, there are many types of symbiotic relationships that fungi are involved in. The big six are listed here. So we have mutualism. This is where there's two organisms, and when they interact, both are benefiting from the interaction. So an example is mycorrhizal fungi. We have commensalism, where one organism is going to benefit and then the other one's not really affected. So it's not getting anything and it's not being harmed by the interaction. A great example of this is all of the fungi or yeast that we have on our skin. It's not really affecting us as humans, but it's surviving off of our detritus. We have parasitism, where one organism is going to benefit, the other one's going to be negatively impacted. So these are all of your pathogens, whether it's plant, animal, or fungal pathogens. We have amenualism, where one organism is negatively impacted by the interaction, and the other one is not at all affected. A great example of this is the fungi Penicillium, because it produces the mycotoxin penicillin, which I'm sure many of you are familiar with. The production of penicillin actually kills bacteria. And although the bacteria is negatively affected by that interaction, the fungi doesn't get any benefit from the bacteria's death. It's just kind of something that happens. We have competition, where both organisms are negatively affected by the interaction. A great example are wood decay fungi. If you are splitting your own firewood, sometimes you see these really dark zone lines in your pieces of wood, and that's just two wood decay fungi bumping up against each other. They don't want to compete for space or food, so they put a lot of resources into these zone lines so they can't cross over. And then lastly, we have neutralism. These are really niche saprotrophs and epiphytic fungi. So this is where two organisms are interacting, but neither are getting any benefit or harm from it. Sometimes fungi can grow on the outside of plants and they're just kind of hanging out there.
So to understand where we are, it's always helpful to know where we have been. And it's just worth noting that mycology has really been an overlooked field until recently. And this is surprising considering they're the largest unrecognized group of organisms on the planet. So if you go back, next slide, please, all the way to the 1700s, they were considered plants, 1800s thallophytes, and it really wasn't until the 1900s that fungi were named as a separate kingdom, which we now call kindom. But whenever you went to study them in a university, it would be in a plant pathology class, or potentially a botany class. There really wasn't a state heritage understanding of fungi's primary role in supporting nearly all listed plants, and the Endangered Species Act didn't really even recognize fungi. In the IUCN, while the European Red List had included fungi, that had not yet crossed the Atlantic Ocean. Now, this all began to shift in the 1990s. This forest ecosystem monitoring team developed a survey and managed guidelines for national forests. And soon after that, a team of mycologists and botanists put together 256 rare species list. And then in the 1996, there was a classic meeting between the Northeast Mycology Foundation and the North American Mycological Association. And at that meeting, basically 4 European red listers showed up and they gave stunning talks about how rare fungi were tracked in Europe and species diversity lists were kept for over 30 years. And that really put a fire under the belly of people in this country. Fast forward to the 20th century, led by mushroom clubs and their leaders, starting really out west in Washington, Oregon, California, particularly with lichens, and then in the Midwest in Michigan and Florida and Wisconsin. A fortuitous grant finally brought the red list to town, and after 5 days of meeting, 48 species were added to that. And you can see here this Calabolides pecci, which is an ectomycorrhizal fungi. If you're not familiar with that, in the category of mycorrhizal fungi, it's organisms who go wrap around the root. And these really interesting, were very widespread, and they're now very rarely reported because they coincide and partnered with the American chestnut, which since that blight has decreased in its presence in the eastern hardwood. It's currently listed as data deficient. So fast forward to time we're living in now. It's a very exciting time. We have this basically national organizations like Fundus have formed to broaden the general knowledge of fungi, and we have these East and West Coast rare fungi challenges. So it's an exciting time for fungi, finally. So looking at the, not sure why the picture's not showing up with the cruise, but yeah, looking at the history of our group, basically it had come to the attention of one of our colleagues who was the former assistant botanist, Aaron Marcus, who really kind of invited some of us to the table to basically create a proposal and submit it to the Endangered Species Committee explaining why fungi should have its own scientific advisory group. And this was just after the Bryophyte Advisory Group joined, which if you're not familiar with bryophytes, they're a non-vascular plant, liverworts and hornworts that provide food and medicine. And basically when we were putting together this this statement, at the same time, we were able to reference the seminal paper that was put out by mycologists in Latin America that was really introducing this concept of the 3 Fs and bringing in the funga to have the equal power of fauna and flora in conservation initiatives. Fast forward, we had in Vermont the Hericium Erinaceus. You can see this lion's mane in the top left corner, becoming the Vermont state fungi. These are just a couple members of some of our Fungal Scientific Advisory Group that did form. We have a leadership team, and then we have a larger group that we'll hear more about.
So a scientific advisory group's helping to elevate the importance of fungi among Vermont's community members and perform the necessary tasks of documenting diversity, rarity, and sensitivity. So this group's been working on developing a catalog or a baseline inventory of known species, when it's appropriate, assessing and reviewing the existing species so we can eventually list things as rare, threatened, or endangered, and then providing outreach and education. So starting with a baseline inventory, before we could even think about fungi that needed conservation efforts, we had to first come up with what a baseline inventory would be and what it would mean. And in theory, a baseline inventory should have been a comprehensive list of all of the fungi that has been known to exist in Vermont over at least the last 100 years. But in reality, we're really starting to document that baseline today, when we're already having fungi being affected by introduced pests, land conversion, and climate change. So to try to come up with this baseline, probably 100 years too late, we have looked at regional fungaria to determine which species are known to have historically occurred in Vermont. We then moved online to databases, including Mycoportal, GBIF, iNaturalist, and Mushroom Observer for Vermont and the surrounding New England and New York states, as well as parts of Canada. And before moving on, I just wanted to highlight this photo here. This is just a screenshot of the kingdom fungi observations that have been uploaded to iNaturalist. The darker the orange is, the more reports available. So there's a lot of information online about fungi. So through a Forest Ecosystem Monitoring Cooperative Grant, we got to work with the Vermont Center for Eco Studies on the creation of the Vermont Fungal Atlas. And the link to this is up here. And basically, this atlas was a game changer for our baseline inventory because it took all of these separate online digitized data sets across all of these different platforms and then congregated them together into a public-facing website. So when we started this SAG, we knew that there was 1,165 species of fungi in Vermont. And then through the creation of this database alone, we now know that there's at least 3,826 species that have been observed and reported. Now, this is a screenshot of the atlas, and all of these are interactive. So you can click on the records, you can click on this little mushroom for a checklist of species. If you're interested in specific data sets like the Pringle fungarium, you could find that through going through here, as well as through images or contributors. Now, my favorite part of this atlas, and what I think is kind of the coolest thing, is the search the atlas search bar right here. And you can type in any mushroom name, whether it's in Latin or English, and you'll get kind of a wiki page of them. So here I have two examples. I have one of birch polypore, which is on the left. And as you can see, it will tell you its description, its taxonomy. There'll be a little graph up here that's showing you the phenology, or when you're most likely going to see it in the environment to get the freshest specimens, as well as how many records we actually have in the state, and when our first and last records are. So this is a pretty common mushroom. I'm sure many of you are familiar with this, even if you don't consider yourself a mycologist. And the data just shows that. We have 600 records. And this was a screenshot from December of 2025. So I'm sure that there's more records than this now. Now, if we compare that to something that we have under the Northeast Rare 20, which we'll talk about later, but a mushroom that we're considering kind of regionally rare, at least regionally data deficient, we can see that in Vermont, we have two records. So we have one from 1964 and then another from 2024. Now, prior to this database being created, if you wanted to find information about Squamina, you were going to have to spend probably days looking through old reports, trying to see if anything's digitized on iNaturalist, going through GBIF. It would have just been hard to track these two records down, where just typing them in the search bar, I got this report in 30 seconds. So it's pretty cool. So this at least gave us a pretty good, robust start to our baseline inventory, and we're going to continue to do forays and add to this data set. So now that we kind of have a robust start to our baseline inventory, we're just starting to scratch the surface on the assessment and review of existing species. So the state of Vermont ranks organisms from common to very rare to help prioritize management. That's what these natural community state ranks are on the top. We also know that there's regionally rare fungi through the Northeast Rare 20, and then we also know that there's globally rare fungi through the ICUN Global Red List, and that's what these middle columns are here. So now that we have this more robust baseline, we can start assigning state ranks to these selected taxa. And then for species that we think might be rare, we can go out and perform dedicated searches to move them from a data deficient category up to something that is rare.
So there's definitely some challenges and wonderful opportunities in this process. As Savannah outlined, right, the IUCN has particular protocols, right, that is interpreting population and mature individuals. This is based on like who's in the substrate, what the mycelial networks are fruiting, and so on. But the fruiting bodies are ephemeral, and oftentimes the habitat quality and associated plants as proxies depends on how much rain we get that particular season. So it's very difficult to get definitive data. Oftentimes the species are present, but they're under soil, not visible. As also referenced, that you can have something that is globally secure, but within our state in an imperiled situation. And so right now, Savannah and I and a few colleagues are in a group that is going through those 3,826 species that you saw on the listserv and doing the research and trying to figure out where are they globally and is there a rank for them statewide, or what do we need to do to determine that? The other thing that's worth noting is that the charismatic species are going to be more likely documented versus potentially ones that are as equally present but not documented. And then, of course, there are those that are just rare. So one of the things that we think is an important collaboration is connecting with scientific advisory groups of folks who work with plants, because many of these fungi, in fact, 80% of plants, right, have mycorrhizal partners. And there's a rapidly shifting field going on right now in fungi and many other categories of organisms due to the DNA capability. And so it used to just be that you would identify things morphologically, and we're finding now that even now, species that might look morphologically similar are actually different when we get to the DNA. So this is dramatically changing what we're knowing and learning, and wonderfully helpful and also challenging in terms of updating old records and being able to keep everything up to speed when it's changing very rapidly. So we are working within this model that is recognized. Maybe many of you in this call recognize the sort of naturalist Bible in Vermont, right, of wetland, woodland, wideland, that is based on this conservation design about these natural communities, right? There's 97 natural communities. A natural community is an assemblage of plants and animals that across a landscape that have similar environmental conditions. And there's 7 upland and 16 wetland, and in each of those, there's currently listed keystone species. And basically, this is a prioritization tool that the state is using to identify and conserve communities that are crucial for Vermont's ecological functioning landscape. And so we're working with these natural communities, and in that, we are starting to be informed in order to determine our foray locations and how our data will be most relevant to these natural communities. So this is an example of one of our spreadsheets where we've triaged the natural communities based on trees that are threatened by current diseases. You have right now, right, the beech blight disease, the beech leaf disease, we have the elm ash borer, and the impending woolly adelgid. And so these communities, obviously you can see red first, then yellow, then green, all have those species. And then on the bottom, those tabs go through the natural communities and color code them. And then we have the locations in the state where they are. So when we're planning our forays, we're trying to, go for the reds and yellows as much as possible. Again, as Savannah mentioned, this is to gather baseline data, which we're over a century behind because these communities are already changing. Many of them already have these trees diseases in them. So we're technically not getting baseline because we're already getting data with the species ridden with disease, but we're doing our best to sort of catch up. Another thing that we're paying attention are these bioindicators like this Usnea hirsuta, known as bristly beard lichen. You can see there's 13 of these in Vermont. This is a fruticose lichen, and it's an indicator of good air quality. And so these are another kind of way of working with fungi to determine environmental health. This is just a snapshot of 2025, the forays that the Fungal Scientific Advisory Group conducted, and all of these are in the natural heritage base, what was found, and which natural communities we visited. It's also worth noting that there are some threats within the fungi community beyond the potential diseases affecting some of our trees and plants. And this golden oyster mushroom is cultivated by many businesses in Vermont. And research indicates, as you can read in this fresh off the presses paper, that when they are in a piece of wood, a forest, that there's decreased native fungi in that area. They're quite aggressive. And so when some of our members learned about this, one of whom is a mushroom cultivator, he quickly changed his protocols of his business, stopped growing it, trying to figure out different ways to contain the spawn that's in the back of the building, and having sort of light education campaigns for other mushroom growers and potentially consumers to maybe consider not buying this mushroom. So that's just another thing to keep track of. We have some exciting other additional fungal research. I'm going to briefly highlight 3 projects, the Woodbury Northeast Wilderness Trust, one at UVM Jericho Research Forest, and a pending project at UVM Proctor Maple Forest. And it's worth noting, this is just a picture of of colleagues that are also part of Fundus. We've recently presented at the Northeast Natural History Conference. So it's just exciting that fungi are beginning to enter the map of more conservation communities like many of you on this call. So this is just from a presentation that Kevin, a colleague, and I gave yesterday to part of the Fungal Scientific Advisory Group, where we conducted an inventory for this Woodbury Wilderness Trust that is part of a much larger wilderness trust, and gave them a report with some recommendations. And we see that as a very beginning protocol of what some members in the Fungal Scientific Advisory Group can do. We're basically doing this through our businesses on the side, because as a scientific advisory group, everything's pro bono, and then we're doing these kind of through our businesses and earning a little for our compensation. But we see this as a valuable tool for anybody who is managing large tracts of land, conservation pieces of land, to basically start inventorying, getting that baseline, and then being able to track how populations change over time. And again, working with the natural communities that are on that landscape. So this particular study is in an incredible wilderness corridor of 6,257 acres, 39 miles of headwater streams. And this core wilderness is in a forever wild easement thanks to the Vermont River Conservancy. And we're also find it fascinating that it's at the divide of 2 different watersheds. And one of the neat things about this site is that it's a classic case study of what Vermont conservation design is really trying to protect and promote. Right now, we only have this 9% of Vermont landscapes of old forests. But when we think about the 30 by 30 and 50 by 50 plans that are in the works, more and more, hopefully that percentage will go up as we can discover these little parcels throughout the landscape. And so we did a literature review of many different methodologies, and we ended up deciding on this intuitive controlled survey, detection, non-detection. We focused on these Basidiomycota and Ascomycota with some lichens and mycoheterotroph plants. If you're not familiar with those potentially jargonous terms, those are 2 different categories of fungi. The terms are basically defined on their reproductive structures. And the mycoheterotroph plants are plants that basically partner with fungi and obtain their nutrients through the fungal networks rather than photosynthesis. And this protocol that we followed does not necessarily capture what's called arbuscular mycorrhizae fungi. That's a category of mycorrhizal fungi that does not have above ground fruiting bodies. And it was a very small inventory. We only did a survey in July, so certainly not something comprehensive. But we thought, again, it could be a model for something, affordable and then could be repeated to be used in future monitoring. So again, we followed the triage that we mentioned of those different categories. So one of the first things we did when our proposal was accepted by the Wilderness Trust was to ask for their natural community map. We then went through the natural community map and sort of triaged which ones were most important to get community population data on based on, the presence of these diseases and those tree species in those ecosystems. And this is just a snapshot of what we ended up honing in on. It was quite close to the road. We were able to get to it. The topography was not too challenging. And it was basically the hardwood, hemlock, northern hardwoods, the northern hardwood forest, the red spruce cinnamon fern swamp, and then the red spruce northern hardwood forest. So our data was basically, a very small amount. We had 49 observations, 28 of them made it to research grade, which is about 57%. And you can see we walked a very small distance in those 5.5 hours, and you can see the trail kind of matching where our points are. And the largest amount of species were found in the northern hardwood forest. So recommendations that were in the report we submitted, and again, we're just sharing this as kind of like a snapshot for you all to consider, like what would it look like to do fungal inventory on lands we tend. We basically recommend, continuing monitoring these natural communities, and that a baseline inventory is much more thorough when you're doing it 3 times a year during those mushroom fruiting periods of spring, summer, fall, and ideally doing that for 3 years to get baseline, knowing that's not a thorough baseline. And then after that, doing this natural community rotation cycle every 2 to 4 years, depending on time and funding. And knowing that with climate change, these natural communities may change. And we already saw that needing to ground truth some of these natural communities, because as those of you who may know, when you're in the forest, there's these mosaic patterns where all of a sudden in the middle of a northern hardwood forest, you might have a tiny area that's, a red spruce cinnamon fern in like a wet spot, and all the tree species and such indicate that. So really important to ground truth. And then also we think the detection, non-detection methodology is fine. And then you can do that for 10 years. But there's many other protocols that would provide much more information, particularly adding eDNA. Definitely DNA sequencing. As I mentioned, morphology is not sufficient for ID. And vouchering is a really important part of our process. And then co-locating permanent plots. Now, this trust happens to already have some permanent plots which they're studying other living organisms, and so adding fungi to those is a good way to go. So just looking at that again in an easier form, like imagine getting that baseline for 3 more years, doing 3 times, in the growing season, then having baseline, revisiting those, and then of course expanding this to include other natural communities and keeping that baseline going for at least 10 years, right? And then you would go through those data sets and be able to determine, or at least get a snapshot of, how the fungal populations may be changing or maintaining, any combination of those possibilities. And while there was nothing incredibly notable in our findings, I think it's neat to just share with you some of the species that we found. And one of them, I'll just say here, this Polyporus radicata, which is actually quite rare in neighboring state of New Hampshire. There's 30 research grade of those in Vermont, but it's imperiled in Alberta It's not yet ranked, so something to keep our eyes on. Looking at this Talipocladium phyoglyces, which is a snake-tongued turtle truffle club, there's 23 research grade observations in Vermont, and they actually have an S2 in Oregon. So again, something worth watching, particularly because of the habitat it grows in. This Cordyceps militaris, which is always exciting to find, 71 species that of research grade in Vermont. Definitely something to track. And this pulmonary lobaria, which actually for some reason did not make it into our species list, though we certainly saw it. One of the naturalists we work with was mentioning what an important indicator it is for air pollution. And while it state is a rare in our state, it's not considered to be something that's very common. It's a G5, but it's actually considered very rare in Europe, and just something to keep our eyes on. There's 446 of them that are research grade in Vermont currently. So yeah, other things to keep in mind is if you are writing grants for having fungal inventories on the land you tend, definitely factoring in DNA sequencing and vouchering. Luckily, there's a new organization called Mycota this year that's allowing free sequencing for Vermont. We're very excited about that. We don't know how long that will continue. And then, of course, there's a lot of other more detailed ways of monitoring. We talked about that yesterday at length, doing transects, doing fixed plots. All of these would provide more data, more information. And yeah, it says thanks, Rick, because one of our colleagues and mentors, Rick Van de Pol, who you'll hear more about his work when Savannah shares about the rare challenges, had kind of looked at our report and our suggestions and also kind of added some of these ideas as well. And I mentioned that, there's this idea of doing transects, which basically provides a whole different data set than fixed releve plots, and the importance of capturing these microhabitats, that could be missed. And that's often where these ectomycorrhizal species are. And then also thinking about plot frequency as well as eDNA, just a lot more information that you would gather. Another project that we're quite excited about, we're actually meeting with them this afternoon. So Kevin Melman, a colleague in the Fungal Scientific Advisory Group, he coordinates a business called Fun Shrooming. Basically, he's a mushroom cultivator, and I coordinate a ecological resilience service, MycoEvolve, offering earthworks education and research. And so outside of our roles in the Fungal Scientific Advisory Group, we are writing small grants to fund these inventories, and then the data all goes back into the database to further Fungal SAG's aims. And so this new study we're beginning this spring is at the UVM Jericho Research Forest. Thank you to the Waterwill and Les Merhoff funding. And the deliverables that we will be offering are to create an iNaturalist project, which Savannah will talk more about the value of that in our data gathering. That will go into the Fungi Atlas and have a species list. We will be doing DNA ID and vouchering, and we'll write a report and recommendations, again, for long-term inventory with hopes of training staff and students so that that's something UVM can start getting baseline and then having long-term data on. We are excited about the prospects of collaborating with the Proctor Maple Research Forest. Our first step being in touch with the research scientist there, Tim, is getting a natural community map created, which is in the works. And then, of course, identifying those triaged areas we've discussed. And there was some data gathered this past year that we're looking at and trying to make sense of, and then writing up grants to long-term research plots, and of course, training staff and students to get baseline and then monitor from there.
So lastly, we're also continuing to work on conservation and outreach efforts. So we keep the Endangered Species Committee informed about rare, threatened, and endangered fungi elsewhere and how it relates to our work. We maintain the state's database of known fungi. When it's appropriate, we'd be proposing rare, threatened, and endangered listings for fungi to the Endangered Species Committee for consideration. And then we're creating educational materials, public programs, and events through which Vermont community members can learn about, interact with, and better understand fungi. So right now, a large outreach effort that we've been working on is the State Wildlife Action Plan, which is the 2025 version. This is the first time Vermont's including fungi in any capacity. And we discussed threats that fungi are facing, including the alteration, degradation, and fragmentation of both fungal habitats and host relationships, as well as just general awareness of fungi in other taxa conservation efforts. As a group, we submitted 34 species, which are on this table right here, and these are either classified as watch list species um, based on their relationships with plant species or natural communities at risk, or might have a limited distribution in Vermont but are observed as regionally rare. So if you're interested in getting involved with fungi, a great way to start is through the Fungi of Vermont program. So this program can be accessed through North Branch Nature Center's website, which is right here at the bottom. You go to their nature programs, and it's the last community science thing right here, Fungi of Vermont program. Or you can take photos directly on iNaturalist. If you go through the North Branch Nature Center website, you will get more information about this project. So how to get started, what is a good observation? Like, what photos do you need to take of what part of the mushroom to make it really easy for somebody else to ID without having the physical specimen? Also, is there any extra features or special notes that you should be putting? So natural communities, we've now added that for this program in iNaturalist. So it will tell you about what a natural community consists of, how you can identify it, and then how you can make that appropriate tag on your observation. And then if you're interested in collecting fungi for DNA or vouchering, there's a lot of really great information on how to do that. You can also do this just straight through iNaturalist. So you are in the field, you find a really cool fungi and you want to take a photo of it. You open the app and take a photo. You can then either put in what you think the identification is if you know it, or if you don't know it, AI or whatever software they're using is going to suggest an identification. And then 3 humans need to agree on an identification for it to get moved up to research grade. So once it's in a research grade status, it's going to actually immediately go into our Vermont Fungi Atlas, which is really cool because we'll be able to use this for conservation efforts. And the coolest part about this project is any fungi observations that are made within the Vermont political boundary is immediately going to be added to this project. So all you need to do is open up iNaturalist and start taking photos of mushrooms. If you're someone who appreciates a challenge, there is an ongoing Northeast Rare 20 challenge you can take part of. So these are 20 charismatic fungi that are thought to be regionally rare and charismatic enough that folks should be taking photos of them, but we just don't have a lot of data on in the region. We are thinking that these 14 that I just highlighted are the ones that are most likely going to show up in Vermont, although it is possible that all 20 could be found here. And these 14 were just pulled out due to past history, as well as climate trends, and just other data that we were able to find on our repositories. If you're someone who's interested in doing forays and learning more about fungi kind of on a hands-on perspective, I direct folks to the Vermont Mycological Society. Many of our members of our scientific advisory group are also members here. And there's forays, classes, monthly newsletters that you can be a part of. So this is a pretty cool partner organization that we get to work with. And with that, we just want to acknowledge FEMC and the Vermont Center for Eco Studies for that grant to help build the Fungi Atlas and then the help in doing so. And then there's a few references here if folks wanted to maybe screenshot for some continued learning. I'll just give it a second here. And with that, we can take questions.
Thank you guys so much. If anyone has additional questions they want to put in the chat, we have a couple I can read off to you guys. The first question we have is, for your records, does it matter if the fungi is observed in a farm or garden setting versus in the wild?
Well, I'd say that we conduct our inventories in the wild. That does make a big difference. Obviously, there's, wild fungi showing up in farms and gardens, and there's also cultivated ones. But what we're really trying to track right now is what is the status of the natural communities.
I would say that all all observations are helpful. That although garden observations are probably not going to be helping with like conservation efforts at this point in time, in the future they might be really important. So if you see stuff, take photos of it, because if it's not us that's using it, there might be other researchers that are relying on that data.
However, if it's cultivated, it's worth noting that
Yeah.
in your INAT entry.
Our next question is, how might somebody introduce appropriate fungi into areas that have been managed that are either included as an active area of restoration or passive areas of revegetation, specifically for wetland restoration?
Yeah, that's actually something I'm involved in right now in some restoration projects. And I think that it's a sensitive topic with commercial mycorrhizae on the market right now. Our research is indicating that commercial mycorrhizae is very problematic. So what I really recommend is if that is something that people are doing, that growing endemic mycorrhizae is the way to do that. We have a guide we published on how to do that, and our recent DNA data indicates there are some changes to the methodology, which we are in the process of amending, of how to do that. And then in terms of saprophytes, yeah, I don't necessarily recommend going in and inoculating areas with any saprophytes that aren't already in that community, just so that we can really support the natural associations.
Our next question is, can you describe what an intuitive or controlled survey strategy looks like? I know you guys had that on the slide of your recommendations for future surveys.
Yeah, I mean, that's actually what Kevin and I conducted at Woodbury, and it is a little bit kind of what it states. So we had parameters around a natural community that we were going to remain in. We agreed to the boundaries of that, and then we each kind of took off in a similar direction and just went to where we felt called, which happened to be, stumps, wet areas, and documented what we saw. And that's kind of in contrast to where you would maybe have defined transects, every 15 meters, you're taking a transect or defined plots.
Is there any equivalent mycological societies in New Hampshire that you know of?
Yes, there is. Matter of fact, our mentor and colleague we mentioned, Dr. Rick Van de Poel, lives in New Hampshire and directs the New Hampshire Mycological Club, which he founded. So I believe you probably can Google that and find that there.
Somebody asking that is interested in the chat. Um, is there any recommendations on when to look for fungi?
I mean, I think that all times are great because even right now, you'll see polypores on the trees, and, older specimens as well, which you can start to learn. But I mean, in terms of like fresh flushes, yes, it's, early spring, there's a whole cadre of species that show up, and then, early summer, midsummer, early fall, are windows. And of course, like if you have a rainy week, the next week you'll often see a lot of fruiting. Savannah, do you have anything to add to that?
No, I think that's great. I think it can vary depending on what you're looking for. I come from a plant pathogen kind of lens, so I'm always out there after we get periods of heavy rain. If we have a really wet spring, we're going to see a lot of our foliar pathogens start acting up. So just kind of following the weather, high humidity, a lot of rain.
Uh, Question, are you looking into any connections of mycorrhizae, fungi, and trees, like in the Finding the Mother Tree? Like
Well,
in
I would
Finding
say in our Fungal Scientific Advisory Group, certainly if we come across ectomycorrhizae, right, which are the type of mycorrhizae that have above ground fruits, if we were in like a cranberry swamp, maybe we see ericoid. Yes, certainly we will document those. As I mentioned previously, the arbuscular mycorrhizae fungi do not have above ground fruiting bodies. Outside of my role in the Fungal Scientific Advisory Group through MycoEvolve and my work as a scientist at UVM, I do have some research projects where I'm working with mycorrhizal fungi to restore degraded ecosystems. And we have robust data and some published papers on that, which I could talk to you about at another time.
That's wonderful. Is there any upcoming projects that you guys want to talk about or draw people to just as we're wrapping up here, um, that they can get involved in through citizen science specifically, or just the general iNaturalist and reporting there?
Yeah, I think if folks could take observations on iNaturalist, that probably helps us out the most. The Vermont Mycological Society hosts, I think last year they did like 25 public forays. Some of those you do have to become a member to participate in, but they're a great group for learning and kind of better understanding fungi as a whole and their role in the ecosystem. So I would direct folks to there. And then lastly, with the Northeast Rare 20, if you're looking for a challenge, trying to find those charismatic, regionally rare fungi would be awesome to get more data on.
Yeah, and I would add to that. So we had a clarifying conversation yesterday among our larger fungal scientific advisory group, really defining that the Vermont Mycological Society is really focused on education for all people, regardless of your entry point, and that the Fungal Scientific Advisory Group is really focused on scientific data. And so our forays are actually not publicized, and they're really for people who already kind of know their fungi-ish, know how to do high quality INAT project photos, and are really okay with us like having a completely geeked out experience of just like gathering and documenting. And whereas the myco societies are much more kind of relaxed and a wider expanse for learning opportunities. That being said, you could go to the mycological forays with Vermont Myco Society and learn those strategies. And once you feel strong in them, usually one or two fungal SAG members will be at those and then join one of our fungal SAG inventories later in the summer if you want to contribute data. Other opportunities to learn about fungi beyond your local mushroom clubs are Northeast Mycological. I'll put these in the chat, and North American Mycological Association. They both have incredible gatherings every year. I believe this year NEMF is in Pennsylvania and NAMA is in Michigan, so a little further than they were this summer, but past summer, but definitely worth going to those because you're with a lot of expert mycologists from around the country. They have multiple forays a day, multiple talks, and microscopy opportunities. So definitely worth checking out.
Those are both great leads for anybody who wants to get more involved. We have another question. For those new to DNA sequencing, how might we get more information about the process for mycota?
Yeah, so I put that in the chat. And I would just say that having Vermont on their map is pretty new. And so I think your best bet is to just contact them directly if you happen to be with a club or group, and have a certain way that you're curious about sending them information. They also have some information on their website, but basically the protocol is not too difficult. You would gather when you gather your specimens after taking quality eye naturalist pictures of substrate location, if it's gilled, all the undersides and the stipe and size, and documenting it well and drying it well. You would then mail specimens to them and will already have an INAT tag that's included in that, and then they would able to do the DNA and have that tracked to it.
Where can we find that list of the Rare 20 challenge? Is that on a certain organization's website?
Yeah, I'll put it in the chat.
It's through Fundus.
So
Yeah.
that's, yeah, slowly adding things.
Yeah, and I can compile some of these too and send them out as links when we have the recording sent out for anyone watching. Great. Well, is there anything else you guys want to add before we wrap up?
Well, I'll just say thank you so much for your interest. We're definitely trying to grow myco-literacy in the community and the implications it has not only for conservation, but also for remediation and other forms of ecological resilience, food and medicine as well. If you do manage a land trust or connected to a wild area that you would like an inventory conducted on, feel free to reach out to us. And either it's something we conclude in our pro bono few forays conducted over the years, and maybe train a team of your people how to create baseline and set up long-term monitoring. And then, of course, Kevin and I would be open to, writing a proposal and being hired. And the more people that we can get involved to know about mycology, the more data we can get, and hopefully the more land we can conserve and help polluted lands return to resilient ecosystems. Oh, and
Well
of
said.
course, using iNaturalist. Sorry. One other thing is just like it's always great once you know how to do that. If you just go out in your backyard or hikes, that's just contributing to the database and it's very valuable. Occasionally, you might find one of the rare fungi or a new specimen.
It's exactly what I was going to say. iNaturalist, and thank you for having us. We're always happy to chat about fungi.
Yeah, thank you guys so much for sharing with us today, and we'd love to have you guys share more whenever and connect our networks.
Thank you so much. All right, take care.
Yeah, take care, everyone.